Yeah, good afternoon, everyone, and welcome. My name is Julian, and I will be talking about the occurrence of purple bones in Icelandic Arctic foxes. So the phenomenon I'm talking about is a very strong discoloration that has first been observed in 2007. And you see here on the picture that this discoloration is very, very obvious, especially when the bone is wet, as it is here in this picture, but also when the bone is dried, it remains obvious. So you don't need any equipment to see it. You can see it with your bare eye straight away. This discoloration is still occurring to this day. This picture here was taken less than a month ago of an Icelandic fox, or the mandible belonged to an Icelandic fox that was killed in April. And to this day, no similar cases are known in Arctic foxes at all. There's one other species which has a similar discoloration, but I will talk about that later. Why study Arctic foxes? Why are Arctic foxes so important? Arctic foxes are an apex predator, and that means that they're on the top of the food chain. So Arctic foxes eat seabirds, for example, puffins. Puffins eat sand eels that are small, silver, snake-like fish. The sand eels eat krill, and krill eats phytoplankton. So if something is wrong, if there's a depression on any of these trophic levels, then we will always also see it in the top predator. And that's why the monitoring of those top predators is so interesting. In addition, there is an effect which is called biomagnification. So assume there's a bit of pollution going on in the ocean, then phytoplankton will collect this pollution. Krill eats, well, more than one bit of phytoplankton, so it will always collect and accumulate this pollution. And then the krill is eaten by sand eel, and the pollution will increase, or the pollutant, whatever it is, will increase towards higher trophic levels. That is all that biomagnification means. And that is why the Arctic fox, being the top predator in Iceland, is making for such a good and valuable indicator species. Another reason to study Arctic foxes is because they're cute, and that is actually scientifically important. Simply because stay with the example of pollution, people are much more willing to change their behavior if it affects a charismatic animal, such as the Arctic fox, rather than a freshwater snail in the nearby stream. So my idea is that this discoloration could mean something very, very important. It could be a very big thing, and nobody has ever looked into it before I did. So I want to give, the higher goal that I'm aiming towards is to find the cause for the discoloration. And I thought I'd start by looking at where and when the discoloration is strongest. So the idea is to quantify the intensity of the discoloration, for which it is necessary to measure the color, and I decided to use photography for colorimetry as a method to measure the color. I chose to select the mandibles. Now, why the mandibles? The reason for that is simply that the Icelandic Institute for Natural History, Náttúrufræðistofnun Íslands, has a very, very high amount of these mandibles. There's a few thousand of them in the collection in the basement, and it just makes sense to start with those. Now, how do they get here? In Iceland, Arctic foxes are still hunted legally, and some hunters collaborate with us, and they send the carcasses here to the institute. Here we take some samples of them, and next to the samples, we also take the tooth, the canine teeth to be exact. These canine teeth are very valuable for age estimation, so they're sent to another lab, and from that we can estimate the population of the Icelandic Arctic fox. Now, as you can imagine, it's not very easy to take out the canine teeth. So instead, it is necessary to boil the heads in water for a few hours. The bone then extends, and it is much easier to take out the tooth. And in that process, we also clean the bones. And it was exactly at that process when, in 2007, someone noticed the first purple mandible, and ever since, notes are made in the sheet if the bone has this discoloration. Now, before I could really get into it, I wanted to make sure that I'm not actually looking at an artifact. So it could be that in one or all the bones that were boiled in a pot at the same time display the discoloration, and none of the others. And then it probably has to do with this single batch and not a natural cause. So I thought about that, and I excluded as many of these possibilities as I could. In addition, I did some statistical tests to find differences between purple and white bones based on the comments that were made in the process of boiling and cleaning the heads. And I found there was a significant difference for location and time, as well as the presence of ear mites, which I will talk about a bit later. For all the other characteristics and body features, there was no significant difference for the mandible dimensions, for their sex, the morph, their weight, all of that did not differ significantly. So my idea is to measure the color using photography and then convert this color into different color spaces. And I use those color spaces to find the best measure for purpleness, and then I classify that. And finally, I can use that to analyze the spatial and temporal distribution of the discoloration. Now let's look at those steps one by one. I decided to use photography for colorimetry over, say, photospectrometry, simply because it is much faster. And as I told you, there's thousands of mandibles down here in the basement. If I did spectrometry, the data would be huge and much more difficult to process. And I wanted to get a very wide spectrum of the samples, and hence decided for photography. What you can see here is the setup that I created. So basically, this box that we see with the gray walls, these gray walls are diffusion fabric, which avoids any core shadows, and the lights that shine on this setup uses daylight lamps. This setup was not moved throughout the entire process, so I get consistent lighting. And with that, I produced about 1,000 pictures, well, 3,000 pictures actually, that all pretty much look exactly like that. I will not get the award for Nature Photographer of the Year for that, but they are very well suited to measure the colors. So I opened them in Photoshop. I manually placed 4 measuring points in each of the frames, and each of these measuring points has 3 values in the RGB color system. RGB just stands for red, green, and blue, and this is the share of the color of each component that adds up to the total. Now, the RGB is a very commonly used color space. All the computer works, all the internet as well, is always or usually in sRGB, and it is quite straightforward. Like I said, you have this triplet of a number consisting of red, green, and blue. If all of these values are 0, the minimum, then we see black. And if all of these values are at their maximum, which is 255 in the 8-bit system, the color is white. And all other colors are a combination of these, of this number triplets. So that's why they're called color spaces, because each of them, or usually they have 3 values, and you can picture them in a 3-dimensional coordinate system. Another possible color space looks like that, and it works a bit different. It has 1 dimension for the hue, 1 for the saturation, and 1 for the brightness, as in value or lightness. And HSV and HSL are actually 2 different color spaces, but the hue is calculated in the same way for both of these color spaces. So the question is, how do I use that to measure a discoloration? I did a statistical analysis of a total of 7 color spaces. So I had a total of 20 dimensions. Again, the hue is calculated in the same way for the previously mentioned color spaces. So total of 20 dimensions, and I used them to find the best measure for purpleness. Now, what is the best measure? It is the dimension that discriminates well between white and purple bones. And I based that again on the binary information that was marked in the process of boiling and cleaning. And I did some statistical tests, and it turns out that almost all dimensions differ significantly. Now that's great. It tells us that purple and white are 2 different colors, and it didn't really get me very far. So I had to look a bit further into it. And this, I know this might sound very abstract to you, so I want to make it a bit more obvious. One color space that I looked at is the RGB normalized color space. It is based off the RGB color space, and it just means that each single dimension is divided through the sum of all dimensions. And what that does is it eliminates the information of how bright a color is, because the sum of RGB, remember, if all of them are at their max value, is white. So the sum of RGB is representative for the brightness. And by dividing all of the dimensions through the sum, you get rid of that information. And as a result, all color points lay in one plane. The image you see here on the right is actually animated, and I want to play that so you can see that all the points lay in one plane that goes diagonally through this color space. That's perfectly fine. It's supposed to be that way, actually. The purple points in this color system represent purple bones, and the gray points represent white bones. And what we can see, I want to play it again so you can see, the purple bones stand out quite a lot. So this is very, very obvious, most notably in the dimension G normalized, which is currently facing away from us. Note, though, that the purple bones actually have smaller values for this particular color space. That is just something to keep in mind. Another color space that I looked at is the LAB color space from the CIE. The CIE is the International Commission on Illumination, and they have created color spaces that are based on the human perception. So how we see color, but not only the sensitivity of the eye, but the whole process that is going on. I don't want to go into too much detail, but these color spaces are based on human vision. And the LAB color space is even more special because it is designed in such a way that it captures differences in color, which is exactly what I was trying to do for my thesis. I did this all in the frame of my master's thesis. So the Euclidean distance, so the distance in a coordinate system between 2 points, simply is the representative for the difference in the color. And this one is also animated, and we see again purple stands out very, very much, and the best dimension, or the dimension in which it stands out most is actually A star, going here from left to right. But also the other dimensions differ quite a lot, and that's why I got a significant difference for the other dimensions as well. Okay, so those two seem to be very suitable. Now, what is A star? Here on the right, we see how the LAB color space is designed, and we see that A star, if the values are positive, represents the share of magenta of the color. So magenta is a good way to measure purple. Most men don't even see the difference between magenta and purple anyways, but all this is a bit more complicated for G normalized. G normalized is the share of green of a color. Now, why can we use green to measure purple? That works for two reasons. First of all, the RGB color space is designed in such a way that green is on the opposite side of purple. So the presence of green means the absence of purple. That only works because the color space was normalized. Remember, brightness does not play a role anymore. Otherwise, white values would always have higher values. White colors would always have higher values for each single of these dimensions. And that's why normalizing it was so relevant. Now, to get a proper meaning out of that, I had to invert it. So high share of green means low purple, and vice versa. So I did a classification, and there's a lot of numbers here on this slide. The only thing to take away is, first of all, I did a standard deviation classification. If you don't understand what that means, don't worry too much. It's not very important. And also, what is important is that how the boundaries were placed and the wave factor. So in A star, we see that low values have a low weighing factor, so has less impact. And for G normalized, low values have a high wave factor, meaning they have a high impact on the final map. So let's look at the result. Spatial distribution, moment of truth. This is what it looks like. And it becomes very obvious that there is a clear focus in the Westfjords. So what this map means is the blue numbers are the total amount of foxes with purple bones per municipality. These borders that we see are the municipality borders of Iceland as of 2018. The blue numbers is the total count, the number of foxes that had purple bones, and the fill color of the municipality is the share. So it's the same, but as a percentage. It tells us how many percent of the foxes from this area had purple bones. The clear focus of that is in the Westfjords, in the top left of the map, to be more accurate, Súðavíkurhreppur, where most of the purple bones originated. Now, what is special about the Westfjords? For Arctic foxes, it is that most of the food in that area is derived from the sea. Therefore, I thought that the cause for the discoloration must be somewhere in the ocean. It is important to know that this map is based off binary data. So this map is based off the information that was present at the institute here, the comments which said if a bone was purple or not. So it's somewhat subjective, right? The person that made the note is the thing that I rely on, and that's maybe not very scientific. And that is why I created these other measures, and that is why I created two more maps, one based off A star and one based off G normalized. I introduced both of them to you before. Now, these were not only the ones that I chose at random those were the color dimensions that were by design, in theory, most suited, but also statistic tests resulted in the biggest difference between purple and white bones for these two dimensions. And we see that the results here are very, very similar. In fact, they're almost the same. So we have a lot of discoloration in the Westfjords, most notably Súðavíkurhreppur, and then there's a little bit going on in the southwest of the country. And the other map had the same. There were a number of foxes which were in the southwest of the country. So that is great. Westfjords is clearly the focus. Now, I talked about the ear mites earlier quickly because there was a correlation between purple discoloration and ear mites. Ear mites are a parasite living in the ear channel of Arctic foxes, and they are pretty much exclusive to the Westfjords. There's a handful which were observed outside of what is defined as the Westfjords, but those were very, very few exceptions. So pretty much ear mites only happen in the Westfjords, and that is also why there was a correlation. It simply happens in the same region. But if we look at the Westfjords more closely, we see that the distribution within the Westfjords is very different between the ear mites and the discoloration. From that, I concluded that it is not the presence of ear mites that dictates the color of bone, at least not of this particular discoloration. Okay, I also looked at the temporal distribution, and as you know, the first bones were observed in 2007, and that is visible also here in this graph. And then there was a little bit going on between 2013 and today. My data just ended in 2019. We're currently working on the data to follow up on that. It is important that this is not a full data coverage. We rely on hunters that send the carcasses to us. So we know that there were purple bones that were from foxes that were killed between 2013 and 2018, but we don't know that there were no purple bones between 2008 and 2012. So we have to be very careful what we conclude from this particular graph. Now, naturally, I had some ideas what could possibly cause this discoloration. Please keep in mind that what follows is not based on thorough scientific research. This is more or less wild speculation, which is suggested by the data that I created. The first thing that came to my mind is that it might be pollution. And then I also thought about watermelon snow. I stumbled upon Echinochrome staining. And finally, I want to talk about the role that sea mussels could play in any of that. But let's look into it one by one. Pollution. We know that the seawater in the Westfjords has high concentration of various chemicals, cadmium, selenium, arsenic, and these are only some of the ones. There are also persistent organic pollutants that accumulate over there in the environment. And yeah, like I told you, top predators have the highest concentration of pollutants because of biomagnification. So it could be that all of that just runs into the Arctic fox and is only displayed there. Another thing to point out is that there was a research which indicated that persistent organic pollutants induce asymmetry in the mandible bones of Arctic foxes. And that happens within a lifespan of a single individual. So this is nothing that happens like evolution over a long process. And I looked at some of the pictures and I believe there was more asymmetry in purple bones, but this is far from scientific. The next thing to mention is watermelon snow. Now, I don't know if you're all familiar with watermelon snow, but there is an algae which is called Chlamydomonas nivalis, and that lives in snow and ice in the water pockets that are in there, and it turns ice and snow pink. It looks absolutely beautiful, as you can see on the picture. You can also have a lot of fun on that. And Drangajökull is the glacier in the Westfjords, and this glacier is affected by watermelon snow. Now, what happens to watermelon snow? If the snow is pink, it reflects less light back to space, so less sunlight, which means it warms up faster and therefore melts faster, which brings a whole cascade of problems with it because it's a self-enhancing process. But my idea was that perhaps the snow is melting this ice. The ice runs into the fjord just south of Drangajökull, which is exactly where the area is where most affected Arctic foxes have been found. So maybe this some color of the algae makes its way into the diet of the Arctic foxes and therefore turns the bone purple. The next thing I want to talk about is Echinochrome staining. Now, I told you in the beginning that there's one other animal which has a similar discoloration, and that is the sea otter off the coast of the US. Now, these sea otters eat sea urchins, and sea urchins have very, very strong pigments, which are called echinochromes, and they have this purple color. So if the sea otters eat the sea urchins, their bone turns purple. And if you look at the picture, the color looks very, very similar to what we observe in Arctic foxes, even though the distribution on the bone is a bit different. I personally think that this is highly unlikely because we have found an Arctic fox which had remnants of sea urchins in its stomach. So we know of an Arctic fox that ate sea urchins but had perfectly white bones. This is not a proof for anything. It just makes it very unlikely. Yeah, the last thing to talk about are mussels. Arctic foxes eat mussels. They're not super happy with them. They have these hard shells, but if there's nothing else around, these mussels make quite a happy snack. Now, mussels also have pigments in their shells, so perhaps there is a similar process of staining going on as the echinochrome staining. That is the first way they are relevant. The second thing is they could be a vector for pollutants. Mussels are filter feeders, and that is why they are generally used to measure the pollution in certain areas. So they filter feed and thereby accumulate pollutants, and that dramatically enhances the effect of biomagnification, or so I believe. But they could also be vector for the pigments. So the glacier runoff from the watermelon snow algae, Chlamydomonas nivalis, could also accumulate in mussels. And when they are eaten, very, very large concentrations of this staining agent make their way into Arctic foxes. To this day, the discoloration has only been observed in Arctic foxes. Why that is, I don't know. There's plenty of glaciers which have watermelon snow, but no animals have been observed with that. So perhaps it's a combination, and all three things have to be present: the glacier, the Arctic foxes, and the filter feeders. It is important to know that the monitoring of Arctic foxes that is happening in Iceland is very, very dense. So it's not very common to have such a good database on such an important and very relevant animal as the Arctic fox. So maybe that is what causes it. Now, for the future research, because I'm still determined to find out what actually caused it, one thing to start is to analyze the feces, and that is happening already. A friend of mine is looking into a number of samples and looking what is found there. And if there's remnants of sea urchins, sea mussels, whatsoever, maybe we can find a correlation. But there's more we can do. We can analyze the stomach contents in a similar way, and I guess you're starting to understand why this slide does not have any pictures on it. But there's many other things we can do. We can measure the absorption of purple bones and compare the absorption maxima with the pigments of mussels, but also with the pigments of the watermelon snow algae. And that is a very, very solid indication going in either direction if we find a result there. And it's rather easy to do. But there's more to do. I told you about asymmetry. That could be done in a more scientific way. There are programs already to be run on a computer which look at the asymmetry, and it's computer based, so you just feed it with the pictures and that analyzes that for you. But also chemical analysis would finally shed some light on the chemical composition and the difference of chemical composition between purple and white bones. But there's plenty of other things that can be done. DNA analysis, maybe all the foxes with purple bones are actually related to one another. If you have money, the list of possibilities is endless. Yeah, that was almost it. Of course, a few people to say thanks to. I want to thank the Icelandic Institute for Natural History for providing me with the samples and a spot to work. I also want to thank my supervisor Esther from this institute and Rannveig from Háskóli Íslands. And big thanks also go out to my examiner Christine, who majorly helped to improve my thesis. I want to thank the hunters for their contribution. Not for shooting the foxes in the first place, but for sending them here, and also Maria from Hafnathing for giving me this opportunity to talk here. And of course, I want to thank you for your attention, and I'm looking forward to your questions. Maybe one of you comes up with the final solution. That would be very nice. Thanks.
Thank you for this talk, Julian. Við þökkum Julian fyrir erindið. Þeir sem vilja senda inn spurningar, þrýsta á hnappinn hérna fyrir í tákninu hérna til hliðar og ég les upp spurningarnar og ef það er í lagi að svara á ensku þá svarar Julian, en annars get ég svarað á íslensku líka. Julian, I have one question.
Yeah.
Do you hear me?
Yes, I hear you.
Yes, since I don't see any questions on the board yet, I want to ask you one question. If you want to compare your ideas about the causes, like from the watermelon snow or the pollutant, what would be the process? How would you do this?
It depends. So, like as you know, there's already someone looking into the feces. So if she happens to find a high, the easiest thing is to compare the sample ideas and see if there's any correlation going on between that. But I'm afraid that it's hardest to exclude the mussels as a potential cause because it's so varied. It's, like I said, it's a threefold suspect, really. So it's a combination of these things, really. To measure the reflectance is, to be honest, what I believe has the highest return on investment, because it's rather simple to measure the reflectance with a photospectrometer and then compare that to a database of pigments which is out there already. And if you have the same absorption maxima, you can conclude on these pigments, and that will be a great help and guide you in a way, or so I hope.
Thank you. We have one question here on the board. Is it only the mandibles or skulls that have the discoloration, or is it the entire skeleton? You can answer that.
I can answer that now, yeah. So the picture that I showed in the very beginning only had was taken less than a month ago, right? And until then, I only knew of the head, so the skull and the mandible, and that was it. And I just assumed all the other bones to be purple as well. But as things are, on that particular day, I saw, while I was taking the samples, I saw that the skull was purple through the skin that is on the skull. So I looked at some other bones. I took this poor fox apart and looked at as many bones as I could. And it turns out that all of them are purple. I took vertebra from the neck. I took paws, legs, and all of that was purple. So I'm pretty sure that this discoloration is based or is found in every single bone of the Arctic fox.
Thank you. We are getting loads of questions now. There's one more. Did you manage to isolate purple bones to specific fjords within the area, or is it widely spread across municipalities? I guess the question is about Westfjords.
That is a very good question, actually, and I have not managed to isolate it. The data I had looked at is very big, so I did not look at the individual entries. Instead, I looked at the entry in which municipality it was found. So I only based this on municipality, but it would be very interesting to do this per fjord, which is very difficult because not for all individuals we have the exact location where the fox was found. For some, we know exactly the GPS locations on like 3 digits after the comma, but on many we don't. So the answer is unfortunately no.
Thank you. There is more. One question here. They're all thanking you for a good presentation. This one would like to know if you found different shades of purple according to age, or do the older bones have darker purple color?
I did not find such a thing, not at all. The discoloration was not, or the strength of the discoloration was not dependent on age. But that is actually one of the reasons why I wanted to look into the intensity. But also, it is important to keep in mind that there's not that many purple boned Arctic foxes. So maybe if I had more, I would see such a trend. And I honestly kind of would expect it to see that, but I could not observe that.
Okay, that is maybe related to the next question. How many foxes or mandibles did you look at? And the person is apologizing if this question was previously answered.
The question was not previously answered, and it is a very good question. In the total data set that I looked at, it was over 4,300 bones, of which 54 were purple. When measuring, I wanted to free my mind from this sort of preoccupation. So I measured at random, which was maybe not the smartest thing, as we realized. So I measured the color of a total of 10 mandibles.
Yeah, with the photography. There's questions in Icelandic. You let me know if you don't want to answer it, but there is one here. I'm going to read it in Icelandic for the record and then answer and ask you in English. Þakka góðan fyrirlestur. Mögulegt að bláu refirnir innan gæsalappa éti meira berjum en hinnir. Það að liturinn sé kominn af krækiberjum og bláberjum sem mikið af Vestfjörðum þegar vel árar. This person is asking if they're eating more berries than the others and if the color could be coming from crowberries or blueberries that are in abundance in Westfjords.
Possible, but very unlikely because the distribution of the discoloration seems very, very random. And in addition, Arctic foxes they do eat blueberries, as far as I know, not very much, but they have been doing that long before 2007. And the monitoring has been going on since the 70s. So I'm pretty sure that purple bones would have occurred earlier if it was blueberries.
Já. Já, ég get líka svarað þessu. Þið heyrðu að hann útilokar það ekki, en ég get fullyrt að þetta ekki litur af bláberjum eða krækiberjum og refir éta mikið af því allt land og þúsundir af þessum refum sem við höfum fengið innsendir hafa verið með fulla maga af berjum og það ekkert samband þarna á milli. There's one more question about, did you see the color on the teeth and on the hard tissue that you kind of scrub off when you're cleaning the mandibles?
On the tissue, I did not see it. So the tissue that comes off the mandibles is boiled, so it has this very dark, or rather dark color, and I did not see any discoloration in it. For the teeth, I saw discoloration on the part that was in the bone, but not the part that stands out. And that is also a very big difference between echinochrome staining in sea otters and what we observe in Arctic foxes, because the sea otters, if it gets very, very strong, also affects the teeth. But the teeth for the Arctic foxes, at least the part that reaches out of the bone, is perfectly white, no matter how purple the skull is or the bones.
Thank you. Here's one interesting question. Would it be possible to identify the color of a living fox bone? Purple is regarded as a color of sadness. Perhaps these foxes have a different temperament or behavior than the other ones. What does the purple fox say? Maybe this one doesn't need to be answered, but there is another one related to it, which is actually, could it be a maternal effect or the diet of the mother that could influence the bone coloration?
Yes, that could be. Thanks for that question. I haven't actually thought about that. The best way to figure that out would be DNA analysis, because the monitoring of live foxes is not dense enough to conclude that. So yeah, that's definitely a very, very interesting thought. I just believe that if that was the case, it must still be from another external factor, because otherwise it would also be observed in other Arctic foxes, or at least in other mammals, and I have not heard of it and have not found it anywhere else. So yes, it's actually even likely that it is the mother that passes it on, but it must still be an external factor causing it. So maybe adding some delay in that.
The last question, because we are going to finish at 4, is that this reminds the person on porphyrosis in cows and pigs. And have you considered inheritance as a possible solution for this? So
I
it's
have,
similar.
And inheritance is not unlikely, because the Arctic foxes of the Westfjords are genetically somewhat isolated from the other Arctic foxes, so that is possible. I never wanted to realize that option, really, because it would not be related to my program that I'm studying in, environment and natural resources. not as much at least. So I was just hoping that it's not some genetic mutation, but it is possible, yeah, very well.
Thank you. Yeah, I don't think we have more time. People can, you put up your email address on the last slide, so people can be in touch if they have more questions. But thank you very much, Julian, and I'm going to give the word to Maria again.